Hello, welcome to another episode of a CNA's Opus. My name is Rich. I'm a certified nursing assistant. Um, Today, I want to continue to discuss the discussion of receptor tyrosine kinase and one in particular, which is the insulin receptor. And the insulin receptor is a little bit unusual or a little bit different than the other receptor tyrosine kinases because it's pre-dimerized. The ligand binding domain of the insulin receptor is already attached. It doesn't need the ligand or the insulin to bind to it first, and then they come together. If I could give you an example, say vascular endothelial growth factor receptor 1, it needs two of them, and then vascular endothelial growth factor, the ligand, will have to come in and attach to one receptor tyrosine kinase, and then it will have to attach to the other receptor tyrosine kinase, which would be vascular endothelial growth factor receptor 1, and then they will come in. And in the ligand binding domain portion, which is extracellularly on the outside of the cell, they will bind. They'll come together and attach. Insulin the insulin receptor does not work like that. It's pre-dimerized. It's already held together before any ligand gets there. So if we start off with the gene right here, this is the insulin gene, and we have two splice variants. We have IRA and IRB. The two splice variants, they don't have enough variation between the two, so they can still work with one another. So there's not enough variation there to cause chaos. They could still work with one another and form the insulin receptor. Before I even go any further, I just want to say that there's 58 receptor tyrosine kinases, and they're grouped into 20 families, and the insulin receptor is one of the receptor tyrosine kinases. So that being said, if I could just show you the structure of the insulin receptor. Up here we have, right here I have coded in green, this is the leucine-rich domain, leucine-rich regions within the ligand binding domain, which means this is the portion where the ligand is going to bind, which is insulin, here and here, both sides. The ligand is going to bind to the ligand binding domain, but within it, it has other conserved protein domains. That's the segmentation. That's what's conserved. That's the segmentation. That's what doesn't change, or at least you don't want it to change. Or maybe I'm not going to get into natural selection, but okay. Anyway, this is conserved. This is the segmentation, and this one right here in green, and this one right down here in green is a leucine-rich region within the ligand binding domain. And if there's a few things I could tell you just about leucine, leucine is one of the branched chain amino acids, and leucine is hydrophobic, so it doesn't have an affinity to water. So those are some details about leucine. The other one right here in blue is cysteine. That's right in the center there is cysteine. See it? Cysteine. And that's a cysteine rich region. So there's a lot of cysteine in this domain right here. A lot of cysteine within this domain right here. Again, within the ligand binding domain. So the cysteine, if I could just tell you a few things about cysteine. Cysteine has that thiol group at the end of its R group or functional group. It has that thiol group, and when two cysteines come together, they can form a disulfide bridge or an SS link. So that's how they can come together there. at the cysteine. Then the next one down here is the type 3 fibronectin. Type 3 fibronectin right there. And one thing I want to say about fibronectin is fibronectin comes in subunits, and you'll see fibronectin a lot with like integrins and used to stick to collagen, in the extracellular matrix that holds cells in its place. But they come in subunits to make up the entire fibronectin molecule, which is going to be important next. That right there is the next one right here is half of a type 3 fibronectin. So it only has half of its subunits there. And then down here, this is the C terminus of the alpha subunit. Remember, the insulin receptor tyrosine kinase comes in the proteins are cut, they're cleaved, and it makes the alpha and the beta. And you need 2 of the alpha and 2 of the beta. And up here, these are 2 of the alphas came together. You'll see right here, alpha subunit. So 2 of the alphas came together here. So you'll see the amino terminus up here, the amino terminus up here, and the C terminus down here, and the C terminus down here. And that's for the alpha subunit of the insulin receptor. But again, it takes 2 insulin, the IRA and IRB, it'll take either or 2 of them, and then cleaved in half to make 1 alpha against 1 alpha, and down here 1 beta against 1 beta. So you're going to need 2 of the insulin proteins cleaved to kind of make 4 proteins to produce the whole receptor. Then here, we'll go a little bit further. Here is, this was half. Now the other half of type 3 fibronectin kind of dimerizes vertically here. So it dimerizes vertically with what's in the beta portion. So this is how it connects with the beta, is via half of a type 3 fibronectin right here. So it goes vertically, kind of like less horizontally and more vertically. Right here it attaches to the beta, and vertically over here to the beta subunit. And that's the amino terminus of the beta. Same thing over here. Then again, here is a whole type 3 fibronectin right here. And then here it goes down to the single spinning alpha helix, which all receptor tyrosine kinases have. And this Y right here is the single letter amino acid code for tyrosine. So we have, you'll see the little Y's right here, and those just stand for the tyrosine residues that are within the receptor tyrosine kinase domain, and also on the cytoplasmic tail of receptor tyrosine kinase. So the little square box right there, that is the domain. Okay, and this right here, the little line right there, that's the cytoplasmic tail. And that's important to note because, as I've said before, the autophosphorylation process of receptor tyrosine kinase is a 4-step process. So first, when the ligand binds here, which will be insulin and insulin, the ligand will bind here, insulin to insulin, and it'll cause a conformational change where these 2 domains will get closer together. See, they're too far apart right now to really activate one another. But once the insulin comes in, it causes a conformational change where these two will come slightly closer together, close enough where they can start sending phosphate groups back and forth. Now here, again, it goes from the receptor tyrosine kinase domain of 1 over to the receptor tyrosine kinase domain of 2, and then 2 takes a phosphate back over to the receptor tyrosine kinase domain of 1. So that's how it activates. That's how it starts right there. That's just the 2 steps, and that involves the receptor tyrosine kinase domains. 1 to 2, 2 to 1, just in the domain portion. That's when things take off, and it excites this region right here. So now it'll start phosphorylating 1 to 2 onto the cytoplasmic tails of receptor tyrosine kinase, which fall outside of the domain. They're on the tails of it. They're just tyrosine residues that are sticking out, and they're adding phosphate. The receptor tyrosine kinase is adding phosphate groups onto it. And the same thing is happening then 2 reciprocates onto the receptor tyrosine kinase cytoplasmic tail, which is obviously just sitting in the cytoplasm of the tail, but still part of receptor tyrosine kinase. One other thing that I want to mention about receptor tyrosine kinase is that within the cytoplasm of the cell, you have receptor tyrosine kinases going, downstream and upstream. They're not just randomly picking up inorganic phosphate groups and putting them onto other receptor tyrosine kinases. They're picking it up from adenosine triphosphate. Receptor tyrosine kinase is picking up a phosphate group from adenosine triphosphate and then transferring it. So it's coming from a specific region. It isn't just randomly picking up inorganic phosphates and moving them around. It's picking them up from ATP, which is your body's main energy source. So anyway, down here is this is what this insulin receptor will first start to do. Is it'll pull up the IRS1 or insulin receptor substrate 1, and here's my little enzyme right here, whatever it's supposed to be. Once this autophosphorylates, these tyrosines get phosphate groups put on them, then that will recruit from the cytoplasm of the cell the IRS1. It'll bring it up to receptor tyrosine kinase, and then once the IRS1 is up there and it's bound to the cytoplasmic tail of receptor tyrosine kinase, then it's close enough to the receptor tyrosine kinase domain where it can start phosphorylating the IRS1 in of itself. Like, it'll just start phosphorylating it. And this is to not actually activate it. It's more of to increase the docking stations. When you phosphorylate a tyrosine residue on receptor tyrosine kinase, it becomes like a docking station for certain proteins that have certain binding domains that will attach to tyrosine, phosphorylated tyrosines. Two of those, well, there's a bunch of them, but two of the more common ones are sarcomology 2 domain and phosphate binding domain are two of the common ones. And phosphate binding domain is the one that is found in the IRS1. So that'll get recruited, and that binding domain and the phosphorylated tyrosine will attach right there, and then the receptor tyrosine kinase domain will start phosphorylating tyrosine residues that are on the IRS1. And the reason it's doing that is it's adding more docking spaces for other proteins to be recruited and activated. If you just left it to the receptor tyrosine kinase cytoplasmic tail, you'd have less. There'd be less docking stations there. So this basically makes the docking space bigger. It recruits this, and this has tyrosine residues on it that the tyrosine domain can phosphorylate all the way around this protein, and that makes the docking space much larger. So other proteins can come up, a lot more proteins can come up and attach and get activated more all at once than if it was just a cytoplasmic tail. It will be a much slower process because it's limited in the amount of space. This kind of like makes the docking space bigger, and that's all I'm going to do today. But then I'll continue on with probably receptor tyrosine kinase and PI3-AKT-mTOR pathway eventually, and Go1 and issues with the heart with it and things like that. So should be some good things coming up. And that's it. Rich, CNA.